Good morning everyone. Welcome to the biology class. We'll continue with the next organelles of the cell, that is the nucleus and the chromosomes. Already we have gone through some of the components of the cell theory, and what are cell theory, what are the structures, Golgi complex, endoplasmic reticulum, why they are included in endomembrane system. Yes. Then we'll go with the next cell organelles, that is the nucleus and the chromosomes. Nucleus, you can look into the chart over here. All the cells have the nucleus, the single nucleus. So this nucleus is the main center of the cell, which also controls all the activities of the cell. So this figure shows you in a cell. This is the structure of the nucleus and external structure, and this is the section of the nucleus. You can see the sections. The nucleus was discovered by Robert Brown. It was discovered by Robert Brown in the year 1848. He described, scientifically, the nucleus is known as karyon. It is known as karyon. Usually, all the cells have only one nucleus, or they contain only one nucleus. So such cells are said to be as uninucleate. If the cell is having only one nucleus, it is known as uninucleate. Take the plant cell and the animal cell. This nucleus, if they are, usually when you observe it, if you observe under the microscope, you will find just as a small dot in the section. For example, if you take a peel of onion peel and stain with saffron stain and observe under the microscope, you will find a small dot-like structure in the box-like cells or in the compartments of the cells. Usually each cell has only one nucleus, but in paramecium it is binucleated. In paramecium it has two nucleus, so we can say that it is a unicellular organism with two nucleus. So paramecium are said to be as binucleated. While in some algae and fungi, you can find two nucleus. There are also two nucleus. Usually the cell is uninucleated, but in paramecium it has two nucleus, so we call it as binucleate. While in some algae and the fungi, there are around 2 to 3 nucleus. But this nucleus is absent in, though it is present in all the eukaryotic cells, but they are absent in red blood cells, pseudotubes, tracheids and vessels. Present in all dividing cells, resting cells, yes, but you can observe in all the eukaryotic cells except in red blood cells. The red blood cells, we say them are enucleated, or if the cell does not contain the nucleus, any cell if does not contain the nucleus, we call it as enucleated cell. It does not have the nucleus. RBC are also enucleated, no nucleus. Enucleated means no nucleus. Red blood cells. These red blood cells, there is a purpose of not having the nucleus. They are helping, they are just function is conduction of oxygen, react with oxygen with the help of a protein called as hemoglobin, which becomes oxyhemoglobin and transports oxygen to each and every cells. As they are not involved in the process of division, they are not undergoing the process of cell division, or they don't require the process of cell division, but as they are involved in transport of substances or the oxygen from one cell to another cell, they don't have the nucleus based on their functions. They don't have the nucleus because of their functions. RBC don't have nucleus. Main function is only transport of oxygen. Tubes. Already in the anatomy part you have heard about the phloem. The phloem fibers. The phloem complexes have phloem parenchyma, phloem fibers, tubes and the companion cells. While companion cells have the nucleuses. Here the tubes are those in the phloem which are help in conduction of food. Phloem is a food conducting tissue. There are tubes. It contains the tubes along with companion cells. Companion cells are associated with the tubes. Tubes at young, but they don't have, when young they have a nucleus, but as they get they don't have the nucleus. So we can say that tubes are also enucleated. Which these are the conducting elements, food conducting elements. The tracheids and the vessels. These are the components of the xylem. That is, the xylem is a water conducting tissue. They help in conduction of water. Main function is conduction of water. So in such structures or in such cells, the nucleus is absent and we call them as enucleated structures. So once again, if a cell has only one nucleus, we call it as uninucleated, and those which do not have a nucleus, we call it as enucleated. And if you in paramecium, there can be two nucleus, while in algae and fungi there can be two or more nucleus. Now coming to the structure of the nucleus. What is the structure of the nucleus? As I said earlier, it was discovered by Robert Brown. The discovery goes with the study of Robert Brown. This nucleus is a double membrane structure. In the plant cell, due to the presence of a large vacuole, it is pushed towards the periphery. In the plant cell, you can see that it is pushed towards the periphery, as the major portion of the plant cell is occupied by the vacuole. But in the animal cell, you can exactly found at the center of the cell. The nucleus is present exactly at the center of the cell. In the plant cell, due to the presence of large vacuole, the nucleus is pushed towards the periphery, while in the animal cell it is present exactly at the center. So we will go with the structure of the nucleus. The nucleus is double membrane organelle with two layers. This is the outer layer or outer membrane known as ectokaryotica. The outer membrane is known as ectokaryotica. This is the inner membrane known as endokaryotica. Is known as endokaryotica, and the space between the outer and inner membrane, this space is nuclear space. Between the outer membrane and the inner membrane, there is a space. That space is called as perinuclear space. It has been said that this space, membrane, it's a double membrane organelles, and you can find 4 components in the nucleus. One is the membrane. The next is the components of the nucleus are membrane, nucleolus, nucleoplasm, and chromatin fibers. So it's a double membrane organelle with outer membrane and inner membrane, and the space between the membrane is called as perinuclear space. The membrane. Inner to the membrane, the fluid material. This fluid material, you can look into this chart. This is the structure of the nucleus. Yes, this is the outer membrane, and this is the inner membrane, and the fluid material inside, which does not contain any chemicals, is the nucleoplasm. Nucleoplasm, which is also known as karyoplasm, is known as karyoplasm. So inside the nucleus, you will find a fluid-like substance inside which does not contain any chemicals. That fluid ground substance present inside the nucleus is known as the nucleoplasm. Yes, or we can say that the membrane, this is the membrane which separates the content of the cytoplasm and the content inside the nucleus. So this membrane, that is in the cell, the membrane is separating the content. This is nucleoplasm and this is cytoplasm. So it is separating the components of the nucleus and the cytoplasm. So the fluid material that is present inside the nucleus is the nucleoplasm, or also known as karyoplasm. This karyoplasm on the inner membrane or at the periphery, it is slightly dense or appear dark in color and is called as fibrous lamella. Is known as fibrous lamella. It is quietly dense. At the periphery, it is slightly dense, or we can say that it is thick and is known as fibrous lamella. Inside, it encloses, this nucleoplasm encloses the 2 components, that is nucleolus and chromatin fibers. The nucleoplasm or the karyoplasm encloses the 2 components. This is the nucleoplasm, where it holds 2 components, that is the nucleolus and the chromatin network. Here the membrane is not continuous. So if you go with that, the membrane, it is not continuous. There are minute space, or there is a pore on the membrane. So the nucleus has pores. You can go with this diagram. nuclear pores are present in the membrane, the pores that is found in the membrane, which help in exchange of materials from the cytoplasm to the nucleus. So this gap is called as nuclear pore. So if you expand this structure, expand and see the structure, the nucleus externally it appears with the pores which are known as nuclear pores. The nucleus externally bear the pores known as the nuclear pores. So if it is expanded, or if you expand and observe under the microscope, you will see this is the outer membrane, the inner membrane, the nuclear pore. If this part has been expanded and observed under the microscope, this is the outer membrane, this is the inner membrane, and this space is perinuclear space, and this gap between the membrane is the nuclear pore. In this nuclear pore, as I said earlier, it will allow the exit of certain substances. It is allowing the exit of proteins and the ribosomes, but prevents entry of most of the substances. Or we can say that it is involved in the movement of the substances between the cytoplasm and the nucleoplasm. So the nucleus has minute pores on its surfaces. Those pores are called as nuclear pores. The expanded figure is over here, where there is outer membrane, inner membrane with the nuclear pore. So in this figure you can see that, yes, in this you can observe this, there is minute gaps here and there. Those are the nuclear pores with an outer membrane and inner membrane. So here is the nuclear pores. The nucleoplasm contains two components, that is the nucleolus and the chromatin network. Nucleolus. This nucleolus is a dark non-membrane particle. Dark non-membrane, it does not have many membrane. It is a dark membrane particles which is mainly involved in the synthesis of ribosome subunits. Ribosome subunits, that is the RNA and the protein. The ribosomes are made up of two components, that is the protein and the RNAs. So this nucleus is involved in the production of ribosome subunits, that is RNA and proteins. So nucleolus is considered to be the ribosomal factories, and as it produces the RNA and proteins, which are the subunits of ribosomes. Hence, the nucleolus is known as ribosome factory. It is known as ribosome factory. So nucleolus is a dark, naked, that is, does not have any membrane, but it is a main component of the nucleus, where it is involved in the synthesis of the ribosome subunits, RNA and the proteins. So these are being released. The subunits are being released which come out of the nuclear pore in the form of ribosomes. So when they come out, some of the ribosomes can be attached to the outer membrane of the nucleus. So you will find that in the outer membrane, see here you can find there are on the outer membranes there are minute spherical structures located. So we can say that the outer membrane is associated with the ribosomes. As they get synthesized in the nucleoplasm by the nucleolus, the ribosomes are being released. As they release, there is only exit of proteins and required chemicals. But this membrane does not allow entry of the substances except the proteins. It will allow the entry and the exit of proteins but does not allow any entry of the substances, not even the charged particles like the ions like sodium, calcium or magnesium. It is preventing the entry of such charged particles. So when the nucleolus synthesizes the ribosome units, they are being released. As they release, they get attached on the membrane of the nucleus. So usually if you observe under the electron microscope, you will see small spherical granular structures present around the nuclear membrane. Those are nothing but the ribosomes. Then again, next, the other inside the nucleus, you will find thread-like structures known as chromatin fibers. This was discovered, the chromatin fibers were discovered by Fleming and another scientist known as Fleming. He observed them and he found that it has two areas. In the chromatin network only, it has two areas. One part when it is stained, when the nucleus is being stained, at certain region the chromatin fibers appeared dark. They appeared dark by absorbing the stain. This part was known as heterochromatin. Chromatin, which stains dark and appears dark as it absorbs stains and is metabolically inactive where there is no crossing over. Such meanings of crossing over and all you will be learning in the next class. But just remember now that heterochromatin is the part in the chromatin threads, in the chromatin fibers, that some region appear to be dark and are known as heterochromatin, which takes or which stain darkly. They appear darkly. While this part which stains lightly are known as euchromatin, which stains lightly. It stains lightly. Observe when it is observed under electron microscope. Right. So the nucleus has four components: the nuclear membrane, the nucleolus, chromatin network, and the nucleoplasm. So here you observe the chart once again. This is the nuclear membrane, nucleolus, nucleoplasm, and the chromatin network. So just a recap of the layers. So nucleus was discovered ground in 1848. And this nucleus you can see when it is in the state of rest, that is when it is not undergoing the process of cell division. So in mitosis there are 4 cell divisions, steps of cell divisions. The first step is interphase, which is said to be as a resting phase, where the cell is taking rest, it does not undergo the process of cell division. So that phase is known as interphase. In the interphase you can observe the nucleus very clearly. So this nucleus has 4 components, that is the nuclear membrane, the nucleoplasm, the nucleolus and the chromatin network. So the membrane has 2. It is a double membrane organelle with outer membrane and inner membrane. The space is perinuclear space. The membrane is not continuous, but there are minute pores in the membrane. These minute pores are called as nuclear pores, which prevent the entry of charged particles. Most of the entry, it will prevent the entry of most of the substances, charged particles and even, but it is involved in the exit of the substances. So we can say that this membrane is separating the cytoplasm and the content of the nucleus. Inner to the membrane there is a fluid material. The fluid material is called as nucleoplasm or karyoplasm. This nucleoplasm holds 2 structures. So nucleoplasm, does not have any chemical components, but it is involved in the synthesis of ribosome subunits. So nucleoplasm is called as ribosome factories. And the 2 structures are nucleolus and the chromatin networks. The nucleolus is a naked, as I said, it is a naked, dark structure, does not have any membrane, but mainly involved in the synthesis of ribosomes. Then the thread-like structures are the chromatin fibers, which are helping in, that is, they have, when stained, they appear in 2 forms or 2 bands. One is dark band and the light band. The dark band is called as heterochromatin and the light band is called as euchromatin. Next, we will go with the functions of the nucleus. What are the functions of the nucleus? Coming to the functions of the nucleus. It is the controlling center of the cell. So it controls all the activities of a cell. It controls all the activities of the cell. The nucleus is the controlling center of the cell. It is controlling all the activities of the cells. Apart from that, the nucleus have the genetic material in the form of DNA. It contains the genetic material. It has the chromatin fibers. The chromatin fibers, that is the DNA, which are the hereditary materials. So it is involved. So this nucleus have chromatin fibers, which in turn have the DNA, which are said to be as hereditary units. The nucleus have the chromatin fibers, that is the DNA, which are the hereditary units and help to carry the hereditary characters from the parents whenever it undergoes the process of cell division. It is also involved in the synthesis of RNA and DNA. It is involved in the synthesis of RNA and the DNA, the nucleic acids. So, in future classes you will come to know in detail. Hope you know, you have learnt in your lower sections. There are three forms of RNA: mRNA, rRNA and tRNA. So, these all are required for the process of protein synthesis or in many other metabolic reactions of our body. And you all know about the DNA, how DNA is very important in the living organisms. So which helps to carry the characters from the parents to the offspring. So the nucleus is involved in the synthesis of DNA and the RNAs. While the nuclear core, it is helping in the exit of the ribosomal subunits, or we can say it is helping in the transport of substances between the cytoplasm and nucleoplasm. The membrane has the nuclear pores. So these nuclear pores help in the transport of the substances, that is between the cytoplasm and the nucleoplasm. And the nucleolus. The nucleus have Nucleolus, which are considered to be the ribosome factories of the cell. So nucleolus, that is the nucleus, have the nucleolus, which are mainly involved in the synthesis of ribosome subunits. So we call the nucleolus as the ribosome factories of the cell. After the nucleus, we will go with the structure of the chromosomes. So, here you can see that this is a part or a section of a cell where there is the nucleus, the endoplasmic reticulum and the chromatin material. So, inside the nucleus, there are thread-like structures as discovered by Fleming. They are known as chromatin fibers or chromatin thread-like structures. Especially in the process of cell division, when the cell divides, when it is undergoing the process of cell division, the thread-like, the chromatin fibers, all the chromatin thread-like fibers condenses and become thick and short structures known as chromosomes. They become the thread-like chromatin fibers during the process of cell division. It is condenses, it becomes thick and short to form the chromosomes. The chromosomes' complete structure was being discovered by the scientist Hofmeister. Gave the complete structure of the chromosomes. Here, these chromosomes can only be observed, can be used, or we can observe them clearly in the metaphase stage of the cell division. I'll give you a rough idea what are the stages. So there are 5 stages in cell division. First with interphase. Yes, there are 5 stages. One is interphase, prophase, metaphase, anaphase, and telophase. These are the stages of the cell division, where in interphase you can see a very clear-cut nucleus with the chromatin network and all the organelles inside the cell. But when it enters the process of cell division, that is the prophase, in the prophase you will see that the chromatin network condenses to form the chromosomes. If you stain with special stains like methyl blue and caramine, you can observe the complete structure of the chromosomes. So it is clearly visible during the metaphase plate or the metaphase stage of the cell division, where all the chromosomes lie at the center of the cell in an equatorial plane. So the chromosomes are the condensed short structures of the chromatin fibers. So let us see what is the parts or what are the parts of the chromosomes. So it has, you can go with this structure. This is the external feature of a chromosomes. There are 2 rod-like structures. There are rod-like structures which are attached at the center. So we will go with what are the list. What are the parts of the chromosome? Is centromere, chromatids, secondary constrictions, kinetic core, telomere, and helical. You can find in the chromosomes the structures. It is made up of centromere, chromatids, secondary constrictions, kinetic core, telomere, and the pellicle. So one by one we will go with these structures. So the chromosomes, as observed, it has 2 rod cylindrical arms. It is having 2 rod cylindrical arms. These arms are called as chromatids. They are known as the chromatids. So it is having a rod cylindrical arm-like structures which are regarded as the chromatids. Both of the chromatids are held at a point. There is a point. That point is known as centromere. Both the chromatids are held at a point and is known as centromere. This centromere is also known as primary constriction. It is known as primary constrictions. So the 2 arms of the chromosomes, the 2 rod-shaped chromatids are held at the center. That center is known the primary constructions, there are protein complexes. There is a protein complex. This protein complex is known as kinetic core. It is known as the kinetic core, where during the process of cell division, the spindle fibers get attached to the kinetic core. This is a place during the process of cell division, that is at the end of metaphase and the beginning of the anaphase. The centrioles will produce the spindle fibers. Those spindle fibers will get attached to this part, to the kinetic core of the chromosomes. So it is the proteinaceous band surrounding, that is present around the centromere, where it is having the microtubules surrounding it. So this kinetic core is surrounded by microtubules. Then the chromatids, the membrane of the chromatids is known as pellicle. The membrane of the chromosome is known as the pellicle. Why? You can see at some places, see this figure. You can find there is a knob-like structure. The part of the chromatid where there is a knob-like structure, or the chromatid shows another constricted region. This is called as secondary constriction. Chromatids at the one arm or both the arm, it is having another constriction and another narrow region. That region is called as secondary constriction, and this knob-like structure around the secondary constriction is known as satellite. And such chromosomes which have the satellite are called as sat chromosomes. So in this figure you can see here, yes, these are the chromatids. This is the primary constriction with the centromere. Yes, the secondary constrictions. Secondary constriction, there is a knob-like structure. This knob-like structure is known as the satellite. But in human cells, there are 23 pairs of chromosomes. All do not have the satellite. In that, there are 10 pairs. There are 10 chromosomes which have the satellites. Yes, those are the 13, 14, 15, 21 and 22. These 5 pairs or the 10 chromosomes have the satellite. The 13th chromosome, 14 chromosomes, 15 chromosomes, 21st and the 22nd. So those chromosomes, the meaning of these chromosomes have the satellite, the knob-like structure. This knob-like structure beyond the secondary constriction is known as the satellite. The functions of the satellite is still unknown, but it does not stain. It is cyanoacidothymidine. That is, it does not contain thymidyllic acid. It is free from thymidyllic acid. So those chromosomes which have the satellite, they are known as the sat chromosomes. In the human, it is 13, 14, 15, 21 and the 22nd, which have the satellite and are known as satellite chromosomes. Inside the chromatids, you can find the thread-like structures. These are chromatin fibers, or they are also known as chromosomes. They are known as the chromosomes. Inside the chromatin fibers, there is a thread-like structures known as the chromatin fibers or the chromonema. So, when this chromonema, the chromonema is being pulled out, if it is pulled out from the chromosomes, you can find the coiled structures inside it with nucleosomes. In the chromatin network, there are coiled structures known as the nucleosomes, which on expanded shows the histone protein on which the DNA is being wrapped. The DNA is being wrapped on the histone protein. So, the chromatin fibers, when they are pulled out, you will find the knot-like structures in the chromatin fibers. In that, certain regions are there that complete knot. That knot are nothing but the nucleosomes. So, these nucleosomes, when expanded and absorbed, the chromatin fibers have, it is the DNA is being wrapped. It is wrapped on the 4 histone proteins, which stretches to form the DNA, or we can say that the DNA are wrapped around the histone proteins. Then the membrane of the chromatin is the pellicle. It is the outer membrane of the chromosome known as the pellicle. And the blunt end, this blunt end of the chromosomes are known as telomeres. They are known as the telomeres. It is known as the telomere. It is a very stable part of the chromosomes. The blunt tip of the chromosomes. This blunt tip of the chromosomes is known as telomere. It is very much stable. It does not allow the disjunction. It does not allow mixing or uniting of the chromosomes. We have 23 pairs. All are separate with each other. They just don't unite with each other. That is, they don't fuse with each other because of the telomeres. In case if the telomere is damaged or it get damaged, yes, there can be mixing of the chromosomes. The chromatids can get entangled with each other, which may lead to some of the abnormalities of the cell in the future. So, telomere is a very stable, but when it is damaged, it becomes unstable and can allow the mixing of the or fusion of the chromosomes. In turn, or in ways we can say that telomeres prevent the mixing, the fusion of the chromosomes with each other. So, this is the structure of the chromosomes. Based on the position of the centromere also, the chromosomes are divided into 4 types. So, once again we will go with the structures. So, each chromosome have rod-like structures which are commonly known as arms and are known as chromatids. Are known as chromatids or usually we call them as the arms of the chromosomes. So these chromatids internally have the chromatin fibers, that is the chromonemma. This chromonemma shows the coiled structures of DNA with the histone protein, that is the histone protein. The thread-like structure, the chromatin fibers, the chromonemma contains the histone protein on which the DNA is being wrapped. Then the two arms are held at the center at a point and it is known as centromere, and the band of protein, a band of protein surrounding the centromere is known as kinetic core, to which the spindle fibers get attached, and the kinetic core is surrounded by the microtubules. The chromatin fibers shows two components, that is the euchromatin and the heterochromatin, and with the DNA wrapped with the histone protein. That here the chromatids at certain regions shows another narrow region known as the secondary constriction, and the part beyond the secondary constriction are known as satellite, and those chromosomes which have the satellite are called as sat chromosomes, while the plant end of the chromosomes are called as telomeres, which are very much stable. But if they get destroyed, they help in fusion or uniting of the chromosomes. Now, during cell division, what happens is that the spindle fibers get attached to the kinetic core, and you can find that the chromosomes take 4 different shapes, or it appears in 4 different manners. So there are 4 you can observe. Based on the position of the centromere, we can observe 4 forms of chromosomes. So we'll see what are the types, those types of chromosomes. During the process of, so generally if you draw the diagram, if you observe the diagram, this is the centromere and these are the chromatids. These are the chromatids. So there are 4 types of chromosomes, or based on the position of the centromeres, chromosomes are classified into 4 types. Okay, you can just look into this chart over here. This one is known as metacentric, this one is known as submetacentric, and this one is known as acrocentric, and the 4th one is not being displayed, which is known as telocentric. If the centromere is exactly at the center, if the centromere is exactly at the center and the 2 arms that is the chromatids are equal in their length. Centromere at the center and the 2 arms equal in length. Usually we will see this type of chromosome as V-shaped. We see it as V-shaped. Such chromosomes are called as metacentric chromosomes. Yes, if the centromere is exactly at the center and the 2 arms are equal, and you can observe that they are V-shaped, somewhat V-shaped, we call it as metacentric. Sometimes the centromere centromere is not exactly at the center. It is away from the center. It is not exactly at the center where there are two unequal arms. They does not have a equal arms. It is unequal arms and usually appears like J shape. Two unequal arms. It is not exactly at the center. The chromatid and the chromosome. In the chromosome, the chromatids, they are not, the arms are not equal with each other. They do not have equal arms. Such are called as Metacentric, which slightly appears to be L in shape. L shaped. This, you can see that this is the centromere and these are the arms. The centromere is not exactly at the center and it shows the arms are not equal with each other. While in the third type, there is a centromere. Look to the figure. These are the chromatids. You may observe that the chromatids, the chromosomes here, the chromatids are, see, one is too long and one is too short. See, in this you can see that this part of a chromosome have a very short chromatid while one is having a very long chromatid. Or if I make it clear, at one side it is having a short chromatid and other side it is having a very long chromatid. It appears to be J shape and such are called as acrocentric. The centromere is in such a place that one arm is very short and the other arm is too long. Such one we call it as acrocentric. While in some you will find that this is the centromere and this is the chromatid. There is in this type there is no chromatid on the other side, or there is an absence of an arm. There is no arm at one side. You will find only one arm or one chromatid, yes, which appears to be I shape and is said to be as telocentric. So, based on the position of the centromere, they are classified into four types. First one is metacentric, that is, if the centromere is exactly at the center and the two arms are equal in their length, they are of equal size, it is called as metacentric. While submetacentric, the centromere is slightly away from the center. It is not exactly at the center, yes, so that is called as submetacentric. And in the third type, where you will see very long arm and a very short arm, that is known as acrocentric. In some, it is in the cells you can find that the chromatin has only one arm. There is absence of another arm, that is the telocentric. So based on the position of the centromere, they are classified into four types. Then if you go with the functions of the chromosomes, what are the functions of chromosomes? These chromosomes are the vehicles of heredity. They are the vehicles of heredity. Yes, help in carrying the characteristic features from the parents to the offspring. They are the vehicles of heredity responsible for carrying the characteristic features, the genetic characteristic features from generation to generations. So chromosomes are regarded as the vehicles of the heredity, and moreover these, when in the process of cell division, they are involved in the synthesis of DNA and proteins, which are very much required for the metabolic activities of the cell. So during the process of cell division, these chromosomes are very much active during all the 5 stages of cell division. They are involved, they are helping, or they are involved in the process of many components of the cells. In that, the main components are the DNA and the proteins. So the chromosomes, apart being the carrying the hereditary characters, they are also helping in the synthesis of the DNA and the proteins. And this centromere will also help in transferring the characters. While you can say that these chromosomes, as they contains the genes, the chromosomes synthesizes the DNA, in turn it has the genes. So genes are responsible for the particular character of an organism. So these are the 2 components of this period session, that is the nucleus and the chromosomes. In the next class, we will go with the ribosomes. Many times you have heard about the ribosomes. Yes, so we will go with the structure of the ribosomes, the centrioles, and how cilia and the flagella in the remaining class, with the differences between the prokaryotic cells, eukaryotic cells, then differences between the plant cell and the animal cells. You are clear about the prokaryotic cells. Now eukaryotic cells, you are all studying the organelles. So you will be clearly understanding the differences between the prokaryotic cells and the eukaryotic cells. So in the next class, we will be dealing with the other cell organelles and with the last conclusion of the plant cell and the animal cell with the diagram. Thank you. Next class, we will go with the other cell organelles.